PCR. Assalamu alaikum, everyone. Yesterday, we started discussing the principle of the PCR, and we discussed the cycling principle of the PCR, and that each cycle consists of 3 steps. Today, inshallah, we're going to learn what happens at the molecular level, what happens during each cycle, for example, and each step within the cycle. Now, this figure summarizes the major steps in PCR and what happens during each step to the DNA, to DNA molecules that are being amplified. Okay? So as you can see here, we just can see the amplification of one DNA molecule during 3 cycles. So here we have cycle 1, cycle 2, cycle 3. So as you can see, at the end of cycle 1, we have 2 DNA molecules. At the end of cycle 2, we have 4. And at the end of cycle 3, we have 8 DNA molecules. So if we apply this equation to this example, so the number of DNA molecules after 3 cycles of PCR equals the number of DNA molecules we start with, which is 1 here, multiply 2 to the power number of cycles, which is 3, which means we have 8, and this is what we can see here. Okay? So let's see what happens during each step within the cycle. So cycle 1 consists of 3 steps, cycle 2 consists of the same 3 steps, cycle 3 consists of the same 3 steps, and these cycles are repeated between typically 25 to 35 times. Okay? Some PCR application requires more, some PCR application requires more cycles, some PCR application requires less. But typically, it's between 25 to 35 cycles are used. Now, typically, denaturation is done at a temperature between 94 to 98 degrees centigrade. The annealing temperature is more variable because it depends on the sequence of the primer. So we have annealing between, let's say, approximately between 50 to 65. And extension typically is done at 72. Okay. And as you can see, we have, let's say, a segment within the DNA or a sequence within the DNA we want to amplify. So if we zoom in, this is the target sequence. We want to amplify only or to make millions of copies of this DNA segment or this target sequence. Okay, so our template DNA would be genomic DNA. Okay, so typically we isolate genomic DNA. Okay, and again, just as a reminder, we put this genomic DNA inside the PCR tube. Okay, so this genomic DNA represents the template DNA. We call it the template DNA, and we add it to the PCR tube. Of course, in reality, we will not have just one only genomic DNA. We will have maybe thousands or hundreds of thousands of the same genomic DNA, which we will add to the PCR tube. So normally we do not have only one, just like in this example, one DNA molecule. No, we will have maybe hundreds or thousands or hundreds of thousands of the same template DNA molecules. Okay. And just a quick reminder, we have to add also, so we add DNA template, which might represent a whole genome extracted from maybe a human cell or any other type of organism. We need to add a DNA polymerase. Primers, usually we have two primers. We have forward and we have also reverse primer. Nucleotides, DNTBs. N means here A, C, G, and T, so nucleotides, and a buffering system that will help the DNA polymerase to do its function. Okay? So we mix all these 5 components inside the tube, and now we take the tube and put it inside the thermal cycler or the PCR machine, and now we're going to see what happens inside. So the first step during each cycle will be called denaturation. The 2 strands are separated from each other. So we're going to only show you, of course, during denaturation, all DNA will be denatured. So both strands, so here we have 2 strands. The 2 strands will be completely separated from each other in all the DNA we add to the tube. But here we are only showing you the separation of the DNA that represents the target sequence. Annealing, during this step, primers will anneal to the target sequence. Each primer will bind to one of the strands, and both primers will not bind at the same location. Each primer will bind to one end of the target sequence. Okay, so these primers are important because they provide a double-stranded region with a free OH group that could be recognized by the RNA polymerase. So now the RNA polymerase, sorry, what I'm saying, the DNA polymerase, and typically we use Taq polymerase. This is the name of the polymerase we use. The Taq polymerase now recognizes the OH group and binds here and binds here. Okay. Next is the extension. So the polymerase start building new strands, and now we have 2 DNA molecules at the end of cycle 1. In cycle 2, so now then at the end of cycle 1, we have 2 DNA molecules, and now we have to go back in cycle 2 and start with denaturation, then annealing, then extension. So these 3 steps are repeated every cycle. So now at the end of cycle 2, the 2 molecules in cycle 2 will be separated. So now we have 4 single-stranded DNA molecules, then annealing, extension, and we end up with 4. In cycle 3, the 4 double-stranded DNA molecule will be converted into 8 because the 4, they have they are made of 8 single-stranded DNA, which could be used as templates in cycle 3 to make 8 double-stranded DNA. Okay. So far we have discussed DNA cloning. We have discussed PCR. And both techniques, or both sorry, something wrong. So we can carry out DNA amplification مضاعفة ال DNA amplification using DNA cloning or using PCR. If we go back to this slide that shows how cloning happens here. In order to do cloning, we need resection enzymes and plasmid, and we cut the plasmid with the resection enzyme. So now we have, let's say, an open plasmid like this. This is the rest of the plasmid. And now we have to cut this fragment with the same resection enzyme to generate compatible sticky ends. So my question is that how can we get a DNA fragment that has two restriction sites on both ends? Where do we get this fragment that we use for cloning? We can obtain this DNA to be cloned by PCR. So the idea here is that you could have like a genome, a very long genome, okay? Very large genome, okay? And you want just to amplify this target sequence from the genome. You want to clone this target sequence. So you cannot just cut this whole genome with the resection enzyme, because you will get like millions of fragments, and you never know, maybe your resection enzyme will cut in the middle of your fragment or in the middle of the target sequence. So the way we obtain this insert is that we could do PCR, okay, using these primers that will bind specifically to your target sequence. And this PCR will produce copies of your target sequence you want to clone, like this. Millions of copies, and then you can cut them with resection enzyme. But you have to understand that naturally your DNA that you want to clone might not have two resection sites on both ends. So sometimes you have to put artificial resection sites. How? By using primers that will have a resection site. So primers, these primers are unique. Unique means فريد من نوعه. These primers will only bind to your target sequence during the annealing temperature. They will not bind anywhere in the genome. They specifically will bind to your target sequence. They are designed So typically they are single-stranded, these primers. Typically, of course, typically means most of the time, they are 21 nucleotides. Okay? Typically, they are between 18 and 30, but most of the primers we use are 21. And you need two, okay? Because these two will allow the amplification or the synthesis of two new strands. So now, how can we can use primers, and we can use them to add restriction sites on both ends of your PCR product that you want to clone. How? So you can obtain fragments that you want to clone using PCR, and then you cut them with restriction enzyme, and then you cut the enzyme with the same restriction enzyme, and you combine them together. Now you have a recombinant. Let me show you how we can use primers and PCR to introduce restriction sites on both ends of a target DNA sequence that you want to clone. So imagine if we have, let's say, a very large genome, and you are interested in, let's say, cloning a fragment here. So here you have sequence or target sequence to be cloned. You want to clone this sequence. Okay. If we zoom in here, and we could write some, you have, let's say, some of the DNA, this sequence. I'm gonna use random. I'm gonna just write a couple of nucleotides, not the whole sequence, just to make my point. Okay, and then we have the rest of the sequence. Then we have a couple of nucleotides, maybe here. Then we have the rest of the sequence, the rest of the genome. Okay. So again, this is my target sequence from here to here. I want to amplify it, and I want to clone it. I want to make more copies of it using the PCR, and then I want to clone. Now we have to use primers, two primers. And both primers should contain also, they should do two functions. So the primers I'm going to use they must do two functions. First, they must bind to my target sequence. Okay. Also, I want these primers to introduce restriction sites on both ends. So in this case, we could use the following two primers. I'm just gonna write a primer that is maybe 7 nucleotides long, but typically primers are longer than that. They are between 18 to 30 nucleotides. So primers are short, single-stranded DNA molecules. So I'm going to write the sequence of the forward primer from 5' to 3'. Sorry. Let me just add a couple of nucleotides here that are not in the target sequence, just to emphasize something very important. Okay. And here we have the rest of the DNA, the rest of the DNA, the rest of the DNA. So let me write the forward primer. Forward primer. So this is the sequence of the forward primer. Okay. And then the reverse, same thing. So forward أمامي, reverse الخلفي. So again, maybe something like this. We have here C, T. Okay, so these are the two primers that that could be used to amplify the target sequence from here to here. Okay, and between here we have some nucleotides. Maybe I should use dotted lines to indicate that here we have many nucleotides and number of nucleotides. Okay. And now I'm going to show you how these primers will anneal to their target sequence. So the first step in PCR is denaturation. So these two strands will be denatured from each other like this. So I'm going to just write the sequence like this, C, 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 and here we have G, 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 and from the other end we have T, A, oh, okay, let me make them denatured like this, A, T, A, and then the rest of the sequence, then here we have T, A, T, the rest of the sequence. Use the pink color to show T, and here we have G, A, T, T, C, G, C. Okay, and this is the complementary strand. The rest of the sequence, the rest of the sequence. Now, I want you to pay attention to this sequence here and here. This sequence, A, T, T, C, it's a restriction site for the restriction enzyme EcoR1. For the restriction enzyme called EcoR1. So now, during denaturation, the two strands are separated from each other. So after denaturation, we have what? Annealing. So now we are done with denaturation and now annealing. The two primers will bind to the target sequence. So this sequence of this primer is complementary to the sequence. So the primer will bind like this. So this region of the forward primer will allow the forward primer to bind to the bottom strand like this. Look at the sequence of this primer, 3' end, but also the primer contains this sequence. So this sequence, the sequence of this primer from here to here is complementary to the target sequence from here to here. The sequence of the primer from here to here is not complementary to any sequence on the DNA. So the primer will bind something like this. We have this part will not bind. It will be something like this. Okay? Now notice the reverse primer will bind to this region of so here we have a G, right? Here we have a G. So G, T. Oh, it's a C, sorry. For the reverse primer will bind to the top strand like this. So here we have G, I'm starting from here, A, T, 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 A, and this is the 3' end. Okay, so if we flip this one, and then we have the nucleotides in the yellow color, they will not bind, so they will have C, T, T, C, T, T, A, A, G, A, T, A, T. So it's 5'. So now, as you can see, the primers will bind, and following annealing, there is extension. So after annealing, we have extension. So after extension, the polymerase recognize this double-stranded DNA with free OH, double-stranded region with free OH. So the polymerase will come and bind. So the DNA polymerase binds here, and a DNA polymerase will bind here, or the tag polymerase. And what happens is that the polymerase will start extending the two strands like this. So let me just use color, and then here we have a G, a C, a G, an A, a T, and it will continue. Okay. Same thing here. The polymerase will make a strand, and then here we have a T, A, T, T, C, A, and it will continue. And after several rounds of, extension, so this is in just one cycle. And if these denaturation, annealing, extension repeated many times, let's say 25 times, using these primers, at the end, what we will have PCR products like this. I'm not gonna write the sequence. I'm gonna show you millions of PCR products, and all these PCR products on their both ends, they have restriction sites. Okay. So after PCR, you will get millions of copies of your target DNA made or produced, and all these copies, they have on their both ends double-stranded restriction sites. So these are, the yellow are restriction sites. So EcoR1, for example, restriction site. And now you can take these. So again, here we have billions or millions of copies of these amplified target sequence, and they have restriction sites on both ends. And eventually, you can take these restriction fragments, PCR. These are, by the way, they are called PCR products, نواتج PCR. They are all taken, and now they could be used, they could be cloned. So these are the PCR products. We just saw how they are made. And these PCR products, they contain restriction sites. So you cut with, let's say, EcoR1 restriction enzyme, or any other restriction enzyme that have its restriction site on both ends. You cut the plasmid with also restriction site, the same restriction enzyme. So this is a plasmid. Alaikum salam, Doctora. It's okay. So a plasmid like this. This is the cloning vector. Can be also cut with the same restriction enzyme. And now we have sticky ends here, sticky ends here, sticky ends. So these are all sticky ends. And we mix them together and we use the DNA ligase enzyme to produce a recombinant plasmid. Now, and then we take the recombinant plasmid and we add bacteria. Let's say billions of copies of bacteria. Not copies, billions of cells. And then we do transformation. Now notice here on this figure, these plasmids we use for cloning, they have genes that make the bacteria resistant to antibiotics. So antibiotics مضادات حيوية. So antibiotics are chemical substances that are usually used to kill bacteria. But this plasmid is carrying here, this region, for example, this is antibiotic resistance gene. So this is a gene that allows the bacteria to become resistant. Resistance means مقاومة. So this gene, if it's inside the bacteria, it will make the bacteria resistant to a specific type of antibiotics. So the bacteria that will have the plasmid will also have the antibiotic resistant gene, which means the bacteria will become resistant to certain types of antibiotics. So now, we could select the bacteria that have our plasmid by growing the bacteria in the presence of the antibiotic. Any bacteria that does not have the plasmid will be killed by the antibiotic. So here, we have to add antibiotic. So the antibiotic resistant gene, it's called ABCR. ABC stands for antibiotic. This is just my abbreviation. So this bacteria and this bacteria, their phenotype is ABCR. ABCR. ABCR superscript R means antibiotic resistant. مقاوم للانتيبيوتيك. It cannot be killed with the antibiotic. ABCS means antibiotic sensitive. Sensitive حساسة and can be killed with the antibiotic. So this bacteria is ABCS because it does not have the gene that makes the bacteria, this gene that makes the bacteria resistant to the antibiotic, and this gene is found in the plasma. Okay? So we can, after adding the recombinant DNA plasmids or after making cloning and we have a recombinant DNA plasmid, we could add it to the bacteria. Now, this bacteria, before we add the plasmid to it, what do you think is the phenotype of this bacteria? Is it going to be ABCR or ABCS? This bacteria, before adding the plasmid to it. What is the phenotype of this bacteria? What is the phenotype? Is it going to be ABCR or S? ABC, not R, no, S. So it's already sensitive. It is, before we use it for transformation, it is sensitive. When we add the plasmid, it will become resistant. Okay? So we add antibiotics to select TR, to select the cells carrying the recombinant plasmid. Okay? All right. So now I want to just summarize what we have just learned. So we can clone a DNA fragment from a genome, let's say. We want to clone this fragment, so we could do PCR to make more copies of this DNA fragment. And then we could cut this DNA fragment with a restriction enzyme. So we have restriction, digestion. To produce DNA fragments with sticky ends. We do the same thing. So we have plasmids inside bacteria. We take these plasmids, and the plasmids are carrying antibiotic resistant gene. So this is basically here we have the target sequence to be cloned. And here we have a plasmid that will be used for cloning. And the plasmid is carrying an antibiotic resistant gene, ABCR gene. We do the same thing for the plasmid. We cut it with the same restriction enzyme. So here we have restriction, digestion. And now the plasmid is opened like this and ready to receive the insert. We combine both the plasmid and the insert using the DNA ligase. Our insert will be joined with the plasmid. So now we have a recombinant plasmid. Sorry. The next step is to transform the plasmid into bacteria. So we use we have a technique called transformation, in which we mix millions of copies of recombinant plasmid. So here we have millions of copies of our target sequence prepared by PCR, millions of copies of plasmids, millions of copies, or maybe thousands or hundreds of thousands of copies of recombinant plasmid. And then we use millions of bacterial cells, and these bacterial cells are, their phenotype is ABCS sensitive. And then what will happen is that we do what we call selection. Selection, actually, we do not see the bacteria. So far we have been showing you like bacterial cells like this. But actually, what will happen is that we grow bacteria on a plate like this. So this is agar plate. And this plate contains media, food, basically for the bacteria, plus antibiotic. So now we will have inside the plate, on top of the plate, on top of the agar plate, what we will have colonies. And each colony consists of millions of cells. So this is bacterial colony, mustamara, contains millions of bacterial cells. All these colonies consist of millions of bacterial cells, and the bacterial cells that will appear on top of the plate, if we zoom in, for example, if we look at one bacterial cell, let's say here, the bacterial cell actually will contain the plasmid, it might also contain my fragment, but also it must contain the gene. So this gene will allow the bacteria to grow on top of the agar media, because the agar media contains antibiotic. So this antibiotic, so this step is called selection, al-ikhtiyar. Selection of cells that have the plasmid. Cells that have the plasmid will be selected because these cells are, so the cells that will grow, all of these cells are A, B, C, R. So they are resistant to the antibiotic. The antibiotic will not be able to cut. So any bacteria that did not take the plasmid will not grow, will not be able to grow, and will die. So only the bacteria that will have the plasmid or my plasmid will be able to resist the antibiotic and will be able to survive and will be able to divide to form bacterial colonies. Okay? Questions so far? So this slide summarizes what we have just learned about cloning using PCR. Okay? So sometimes we clone DNA fragments that contain genes so that these genes can be expressed inside bacteria or eukaryotic cells to produce proteins. Okay, so sometimes we clone a gene. Let's say that we have maybe a human gene, and this gene could be cloned into bacteria, into a plasmid, and this plasmid could be inserted into bacteria, and now inside the bacteria, what will happen? So this is bacterial chromosome, and now we have our plasmid carrying our gene, and now the gene could be expressed. Now, cloning our gene into this plasmid requires that we have to have a promoter. So here we will have a promoter that will ensure the expression of our gene like this. Once the gene is inside, let's say, a bacterial cell or a eukaryotic cell, it depends. So now, when the gene is expressed, now we have proteins. So let's say that this is gene X, and now we have protein X produced inside the cell. This cell could be bacterial or eukaryotic cell. So we have something called bacterial expression systems. So bacterial expression system, basically, we use bacterial expression, or the bacterial expression system consists of bacterial expression, let's say, vectors or plasmids, and bacterial cells in which the plasmid will be expressed, in which the genes carried by the plasmid, bacterial host or bacterial cell. Okay, just like here, this is a bacterial expression system, which means we have a bacterial cell that can accept our recombinant plasmid, carrying our gene, and this gene or the plasmid is carrying a promoter, and the promoter can express our gene. So this promoter must work inside bacteria, because not all promoters will work inside bacteria. So we have to choose a promoter that we're gonna be 100% sure that will work inside the bacteria. Okay. So a bacterial expression plasmid or bacterial expression vector. So plasmids are types of vectors. Okay. So this expression vector like here. So this is a recombinant. Oh wow, one at a time. Recombinant vector. The vector will be carrying or will have a strong promoter. Strong promoter means that the promoter will allow the expression of the clone gene inside the host cell, inside the bacterial cell, at a very high rate. Or it will allow high level of expression or high rate of expression. Okay, so highly active promoter or strong promoter. You need your promoter. You need to make sure that the promoter that you are using will work and will be active. Must work or be active inside the bacteria. Okay. Some sometimes we cannot express genes, eukaryotic genes in bacteria. Do you know why? Do you know why sometimes we cannot express genes from eukaryotic cells inside bacterial cells? Anyone? Do you know what's the difference between eukaryotic genes and bacterial genes or prokaryotic genes? Can you give me a major difference? So eukaryotic genes, they are made of exons plus introns. So bacteria, they cannot remove introns. They cannot remove introns, so they cannot express eukaryotic genes. But sometimes we can express eukaryotic genes if we convert the eukaryotic gene. Okay, so we have a eukaryotic gene, for example. And this eukaryotic gene is made of, let's say, exons and introns. So we have exon 1, exon 2, exon 3. And in between we have the introns. So this is intron and this is another intron. So both are introns. So we can take the eukaryotic gene and remove the introns by using, for example, the mRNA. So the mRNA of the gene like this will contain, so this is the mRNA of the eukaryotic gene, and we could convert the mRNA into something called cDNA. And the cDNA is actually a double-stranded DNA. So this is gene cDNA. So this requires of the use of something called reverse transcription. So reverse transcription uses mRNA as a template. Okay. Of course, not this mRNA, because this mRNA is the pre-mRNA, and then the pre-mRNA will be converted into an mRNA that contains the exons, for example, only. So introns will be removed by RNA processing, let's say. And then we could use this mRNA as a template for reverse transcription to produce a cDNA molecule that it's a double-stranded DNA molecule that contains only the exons. And now we can take this one and clone it and into an expression vector, and maybe clone it into an expression vector that have active bacterial promoter. Because this eukaryotic gene might have a eukaryotic promoter. So this eukaryotic promoter might only work inside eukaryotic cells. So we need to take the cDNA and clone it into a vector, and this vector, and put the gene. Okay, so it might be gene X, but we are cloning, for example, here gene X cDNA. So C stands for complementary. And in the plasmid, we have a strong promoter. And this promoter, if we want to produce the protein inside bacteria, this should be a bacterial promoter. Strong bacterial promoter. So we do not rely on the natural eukaryotic promoter. We have to take the cDNA and put it inside a vector, and this vector, so we clone the gene or the cDNA downstream a strong bacterial promoter, so that this bacteria, we will be sure that this promoter will be active inside the bacteria, and it will be able to transcribe the cDNA, and now we have protein X produced inside bacteria. Okay? Some expression systems are eukaryotic expression systems. So eukaryotic DNA could be cloned into vectors, and these vectors could be, these expression vectors could be used to express the eukaryotic genes inside eukaryotic cells, like yeast khamira. So yeast is eukaryotic unicellular cell, unicellular organism. So it could be used like bacteria to express eukaryotic genes that cannot be expressed inside bacteria. Okay? Sometimes mammalian cells or insect cells, they are also used to express proteins, eukaryotic. So one way of introducing DNA into eukaryotic cells is by the use of a technique called electroporation. So electro means electricity, kahraba. Poration, this is from pores, ثقوب. So in this technique, electricity will be used to make pores in the membrane of eukaryotic cells through which the recombinant DNA or the expression vectors can be introduced into the bacteria, into the eukaryotic cells. Or we could use microscopic needles, very small needles. Needles means, yes, Jude, you are right. Needles, ibar. Okay, but they are very, very small needles. You have like cells. This is a cell, a eukaryotic cell. This is the nucleus. And we could have, for example, a needle, a very, very fine needle, microscopic needle. You cannot see the tip of the needle, only under the microscope. And this needle contains the recombinant DNA we want to introduce. Okay, introduce into the cell. So now this needle can inject the cell, or can be used to inject this DNA into the nucleus. So we have something like this, the needle, and the DNA will be inserted into the nucleus. And then genetic recombination will ensure that this DNA will be incorporated into the DNA inside the nucleus. Okay? You have to understand, just like Jude said, sometimes we cannot express certain genes, certain eukaryotic genes inside bacteria, not because of introns, but also some eukaryotic proteins require post-translational modifications. So some eukaryotic proteins in order to become active, they need post-translational modification. They need to receive some chemical groups, and bacteria, they cannot carry out post-translational modification. So the protein will not be active. It will not be produced in its active form. Okay? So not only the presence of introns, because we can remove introns. So basically we can remove introns by making cDNA, but still some proteins might require post-translational modification. So I will stop here.